Welcome everyone to this educational channel on Biology Form 4 and Form 5. My name is Janet and I'll be teaching you biology. So let's study biology together. These lessons are taught with clarity and I'll be using creativity and critical thinking to enhance your learning. To receive notification of new biology videos, please click the subscribe button below to subscribe. Here are a few suggestions on how to use these videos to maximize your learning. Firstly, go through the whole video and listen to the explanations carefully in order to understand the concepts, the naming of parts, mechanisms, importance of certain sections in the exams, and so forth. Once you've understood, then you're ready to jot down the important points if your notes are incomplete. Or you can jot them along the way as you are listening to the video. So if your school notes are incomplete, now is the time to make them more complete so that when the exam comes around, you do not have to go searching for good notes again. Thus, add the missing points to your notes from the video notes. You can screenshot the important points on your phone and copy them down from the phone. Also copy the overviews, which are the summaries or the main gist of all the lessons, as these overviews are very useful and some of them are not found in the books. Next, number 3, take note of the techniques of answering essay and structured questions, the common mistakes made by students, and other suggestions and observations. All these suggestions that I've made are gleaned from my many years of teaching biology and also from the feedback from my students. Furthermore, the notes are also attested to as being of very good quality, as they have been improved through the years, and many students have obtained A+ by using the notes. Fourthly, jot down the acronyms too. These acronyms are not easy to find from books. In fact, there are no acronyms from the revision books at all. And again, these acronyms have been used by my students over many years and they have found them very effective, as I have the opportunity to talk to the A+ students after the SPM and ask them as to the effectiveness of the notes and the acronyms. So they have told me that the acronyms are very useful and they can still remember them to this day. After you have jotted down the acronyms, try to practice using them and testing your memory with them. Then memorize what you have learned also from the notes. Memorize your notes, because this step cannot be done by the teacher for you. That brain belongs to you and you are the one that has to train your own memory. There's no such thing as a poor memory. You just need to train it by reviewing, by repetition, and by testing over and over again. Okay, so memorize what you have learned, your notes, by reviewing the notes. Lastly, to apply all of what you have learned, you need to attempt as many past year questions and other revision questions also. So once you have finished listening to a video, you can try some past year questions related to the topic and see if you can answer the questions. Also attempt all questions in the videos and you can write down the answers as part of the practice and rehearsal for the real day, your exam day. So this is one of the tips given by my A+ students, that you can write down the essay answers and the model answers, especially the very good answers, and that will help you to remember the points to be used in the exam on the day itself. We have already studied the monosaccharides. So in this second video, we will study the disaccharides. So this video is about Form 4 Biology, Chapter 4, Subtopic 4.2, entitled Chemical Composition in a Cell. And we'll be studying Carbohydrates Part 2, that is the disaccharides. So here we have a representative of the Disaccharides, which is the beetroot. Bertie the beetroot. Beetroots contain a lot of sucrose. And he's taking his breakfast, so he has a cake and some sugar cubes. The sugar cubes contain a lot of sucrose, or it is mainly sucrose. Cakes also use a lot of sucrose as one of the main ingredients. Grains contain a lot of maltose, another disaccharide. Then we have sweet fruits, which may contain a lot of sucrose. Milk contains a type of milk sugar called lactose. So these are different examples of foods that contain disaccharides. Let's dive right in and learn their characteristics and how they are formed and broken down. We have already discussed the overview for carbohydrates. So let's go through very quickly again. Carbohydrates consist of monosaccharides, disaccharides, and polysaccharides. The first type of carbohydrate are the monosaccharides, which we discussed in the last video. Now the second type of carbohydrates are the disaccharides. They are complex sugars with the formula C12H22O11. Okay, notice that the ratio of C, H, and O is not 1:1. If it's 1:1, it will be C12H24, but here is H22, and it should be O12, but it's O11. Now the reason for the lack of hydrogen and oxygen atoms, the fact that it has less hydrogen and oxygen atoms for disaccharides, is because when two monosaccharides join together, a molecule of water is removed from the molecule of the disaccharide. So examples of disaccharides are lactose, maltose, and sucrose. As for polysaccharides, we'll study them in the last video. For the learning outcomes, after this video, we should be able to explain the characteristics of disaccharides, explain the formation and breakdown of disaccharides, and we should be able to write and explain the word equations for the formation and breakdown of disaccharides. We have already finished studying the monosaccharides. Let's move on to disaccharides. Disaccharides, the word di means two. Formula of disaccharides. Every disaccharide has the formula C12H22O11. They are also called complex sugars. Examples are maltose, sucrose, and lactose. Their physical properties are the same as that of the monosaccharides, which are the simple sugars. So for these complex sugars or disaccharides, all of them have the same properties. Here, that is, they taste sweet, they are soluble in water, and they form crystals. Disaccharides are formed when two monosaccharides combine through condensation. So let's find out what condensation is. When you see this word D, it means definition. You have to memorize the whole definition word for word. Okay? So condensation is a chemical reaction where two smaller molecules combine or join together to form a larger, more complex molecule with the removal of one water molecule. That means one water molecule is formed in the end on the right side of the equation. Okay, so here we see the process of condensation. One monosaccharide represented by the hexagonal shape plus another one. So the two will join together, but in the process, to form this bond here, one water molecule has to be removed from the disaccharide that is formed. Okay, so that's why it's called condensation. We have studied condensation in chemistry, and it means that it's a condition where water changes from the gaseous state back to the liquid state, and water can condense or form on some cold surface like glass. Okay, so condensation involves the appearance of the water droplets. Likewise, for biology, in biology, when two smaller molecules combine together to form a larger molecule and water is formed, we call this process condensation. Although the comparison with chemistry is not exactly all the same. Okay, the similarity is that a water molecule or water molecules are formed. To make it clearer, let's have a look at the word equation. Monosaccharide plus monosaccharide through the process of condensation produces a disaccharide plus water. Okay, so remember, condensation leads to formation or removal of a water molecule from the product disaccharide that's formed. Here are three examples of condensation. Three specific examples. Glucose plus glucose. When two glucose molecules join together through condensation, maltose is formed. So maltose is a disaccharide and water is removed or formed. Okay. Glucose plus fructose produces another disaccharide called sucrose plus water. And to form lactose, we need glucose plus galactose, another monosaccharide, okay, to form lactose plus water. All these processes are condensation. All right. So whenever you see water molecule formed, that means it's on the right side of the equation. That means it is a process called condensation. Okay. Another observation is that for the formation of all these disaccharides, at least one molecule of glucose is involved. Okay, the second molecule may be different from glucose, or maybe glucose too. If it's glucose plus glucose, it's maltose. Glucose plus fructose, you get sucrose. Glucose plus galactose, you get lactose. From galactose, you get the word lactose. About the breakdown of disaccharides. The breakdown of disaccharides is just by the opposite process. Opposite of condensation is hydrolysis. So disaccharides can be broken down into monosaccharides by hydrolysis. So again, we have to memorize the definition for hydrolysis, as this definition has been tested many times in the exam. Same with condensation. Okay, you need to use the definition to explain the observations and to answer the question. So please go and memorize the meaning of condensation and hydrolysis. Say with me, hydrolysis. Okay, so hydrolysis is a process whereby a large molecule is broken down into smaller molecules. So from large to small, the reverse of condensation, with the addition of water. That means you have to add water. It's on the left side of the equation, left side, and then it will break down the disaccharide into monosaccharides. Now, an example of hydrolysis in the lab is when you have sucrose in a test tube and you add hydrochloric acid, which is diluted hydrochloric acid and contains water also, and heat it up till it boils. Heat it up gently, okay, maybe in a water bath. So hydrolysis occurs whereby the sucrose will be split or broken down, better to use the word broken down, into glucose and fructose. Now, what's the use of the hydrochloric acid or HCl? Okay, it is to make the reaction faster. Okay, but the main reactant is water plus sucrose. We'll look at some examples in the next part. So another example of hydrolysis that takes place is in the body. Takes place in the body when sucrose is digested or hydrolyzed by the enzyme sucrase in the small intestine. So enzymes can hydrolyze the sucrose, can catalyze the hydrolysis of sucrose and break it down into glucose and fructose as part of the digestion process. Let us look at some examples of the breakdown of disaccharides. This is the general formula. Disaccharide plus water through hydrolysis produces monosaccharide plus monosaccharide. Okay, so this reaction is the reverse of condensation, because condensation is monosaccharide plus monosaccharide through condensation gets you disaccharide plus water. So hydrolysis and condensation are the reverse of each other. Right, so normally in the objective questions, they can test you, which process is hydrolysis, which process is actually condensation. So it's very easy to remember. You must look at both sides of the equation. And if the water is on your left-hand side as a reactant, then it's hydrolysis. If the water is a product on the right-hand side of the equation, it is condensation, because water is formed in condensation. To help you remember, you look at this word hydrolysis. Hydro means water. Lysis means breakdown. So imagine that this word is divided into two, hydro and lysis. Hydro before the arrow in the equation, lysis is after. So hydro means the water is at the left-hand side, the front of the equation. Lysis means breakdown. That means on the right-hand side, you will get the breakdown products. Okay, so from this word hydrolysis, we find that water is the reactant on the left-hand side. Okay, and when you see an equation like this, where a complex molecule reacts with water to produce simpler molecules, then that is hydrolysis. Right, let's look at 3 examples of hydrolysis using the disaccharides maltose, sucrose, and lactose. So all these 3 disaccharides can be broken down using water and an acid to catalyze or make the reaction faster. Okay, but the reactant is water. Maltose plus water, hydrolysis occurs to form glucose plus glucose. Sucrose plus water, through hydrolysis you get glucose and fructose. Lactose plus water, through hydrolysis you get glucose and galactose. It's all the reverse of condensation. Here I'm going to give you a special memory tip which I've been sharing with my students for many years. It's very easy to remember the products of hydrolysis of a particular disaccharide. So memory tips for breakdown of disaccharides, important for objective and structured questions. They always ask what is the product of hydrolysis of maltose or sucrose or lactose. Or they may ask the reverse, condensation. If a glucose molecule combines with a fructose molecule, what do you get? What kind of type of disaccharide do you get? Okay, so I'm going to teach you 3 acronyms here to remember. It's very easy and very useful and easy to remember. Maltose plus water through hydrolysis, it will produce glucose plus glucose molecule, 2 glucose molecules. Remember Maggie, M for maltose, G for glucose, G for another glucose. Okay, sucrose plus water gives you glucose plus fructose. Remember sugar free. S for sucrose, G for glucose, and F for fructose. Lactose plus water gives you glucose and galactose. Remember, for lactose, lady, glucose is gu, and galactose is ga. Lady, gu, ga. Not Lady Gaga. Okay, so even though it sounds funny, but most students can remember this because it is something different. Okay, an acronym that's funny. So you can write down all these acronyms in your notebook or on a paper and stick it up the wall. So in case it comes out in the objective or structured questions, you will know the products of hydrolysis of a particular disaccharide. Or even for condensation, you'll be able to predict the results. Okay, so these 3 acronyms were mentioned by one of my students in her own YouTube videos. And if you see these 3 acronyms mentioned by her in the YouTube videos, you remember that this is actually the acronyms that I have thought up earlier, and it's my original invention. What are some everyday food or vegetables or substances that we use and we eat that contain the disaccharides? So sucrose, the sources of sucrose are sugarcane, from where we get the table sugar. So table sugar is actually sucrose. Okay, the one that we use to sweeten our drinks with. Sugar beet is a vegetable that is sweet and red in color, which people make soup with, right? I don't know how many of you have eaten sugar beet before, or beetroot. It's very sweet and red in color. The soup will turn dark red. Sweet fruits also contain sucrose, sweet fruits such as bananas and apples contain sucrose. Maltose is found in grains, and grains are used to make cereals and bread. So cereals and bread will contain maltose. Lactose is milk sugar, so it's found in milk and also milk products like yogurt. An interesting fact is that lactose, sometimes people do not have enough of the enzyme lactase in the small intestine to digest the lactose in the milk. So if they drink milk, they will end up with diarrhea and stomach ache because they will become allergic to milk. That's because their bodies cannot produce enough of the enzyme lactase to digest the lactose in the milk. So they get indigestion or diarrhea, and they cannot drink milk or eat any milk products. In the previous video, we have already stated and explained that the 3 monosaccharides are all reducing sugars because they can reduce the copper 2 ion in Benedict solution to form copper 1 ion, which then forms a brick red precipitate called copper oxide. How about the disaccharides? Are they reducing sugars or otherwise? Lactose and maltose are reducing sugars. That means they will produce a brick red precipitate when heated with Benedict solution. However, sucrose is a non-reducing sugar because it does not reduce copper 2 ions in copper 2 sulfate to copper 1 ions when heated with Benedict solution. So remember, sucrose is a non-reducing sugar. It is not a reducing sugar, in other words. For sucrose to produce a positive result with Benedict solution, that means to form the brick red precipitate, it first has to be hydrolyzed, undergo hydrolysis, or be broken down into its component monosaccharides, which are glucose and fructose. By heating the sucrose with dilute hydrochloric acid, so this acid is meant to make the reaction faster, right? But the actual reaction is between sucrose and water to produce glucose and fructose through the process of hydrolysis. After heating sucrose with dilute hydrochloric acid, we cool the solution and neutralize it with some sodium bicarbonate, and then we test that solution with Benedict's solution. Then it will be able to reduce the copper 2 ion in Benedict's solution to form copper 1 ion because of the glucose and fructose which were formed. They are both reducing sugars. So a brick red precipitate will be formed. The copper 1 oxide will be formed at the end of the reaction. And this is a positive result for reducing sugars. The reducing sugars are actually the glucose and fructose produced through the hydrolysis of sucrose. We have already studied 3 acronyms, which is Maggie, sugar-free, and Lady Guga for the hydrolysis of disaccharides. Here is another memory tip that is very useful to help you remember the 3 examples of disaccharides, which is lactose, maltose, and sucrose. Remember the acronym LAMAS. A llama, L-L-A-M-A, is a South American animal that's used to do some domestic chores, and it's a relative of the camel. Okay, so they're very cute animals and they're also very intelligent. Now, LLA, just remember LA. LA is for lactose, MA is for maltose, and S is for sucrose. You can actually ignore one L because llama, L-A-M-A-S, L-A-M-A is the scientific name for this animal. Okay, the genus is called llama. But never mind, just remember the common name, if you can. Llamas for lactose, maltose, and sucrose. Okay, so that ends the video for today. Disaccharides will be discussed in the last video. And please like, comment, subscribe, and share with your friends. Thank you.